everyone. Welcome to Colloquium. I'm happy to welcome our speaker today, Chris Quinn from UW-Milwaukee. Chris got his undergraduate degree from Rutgers University in biological sciences, and then his PhD from Yale, studying with Susan Hockfield. He is studying neurodevelopment and how axons grow out in neurons in a dish. That's a beautiful process to study, but he had a notion that if he switched to a genetically tractable animal, he might be able to take advantage of the power of genetic analysis and also see how axons develop and how neurons develop in the context of an intact animal where the neurons can respond to their extracellular cues. And so he moved back to Rutgers to William Wadsworth's lab to study these questions in C. elegans, and has been studying them ever since in his own lab in the biology department at Milwaukee. Let's see. So he's studying fundamental questions in how the nervous system is wired using genetic analysis. He continues to study these topics with an emphasis on genes that are implicated in neurodevelopmental diseases and other diseases, especially autism. I appreciate and admire the way that he lets the genetics lead the way and reveal what the genes are doing in neurodevelopment rather than relying on preconceived models. And I'm excited to hear what he's going to talk about today. So thank you for coming.
Thank you, Claire. Great. Good afternoon, everyone. It's a real pleasure to be here with all of you at the Genetics Colloquium this afternoon. And I'd like to start by thanking Claire for organizing my visit here today. It's been a really great opportunity for me both to meet with faculty as well as students, and also to learn about a little bit more about some of the amazing work that's being done here on campus. So my lab really focuses on using C. elegans to investigate some of the basic cellular mechanisms that regulate both normal axon development as well as axon degeneration. And all of the experiments I'll talk about today have been done using this PLM neuron. So it has its cell body in the tail of the worm, and then it extends this axon along the lateral body wall, and it terminates right in the mid-body before reaching kind of the territory of this other neuron is known as the ALM neuron. So, and the other thing about this PLM is the axon actually serves as a touch sensor. So if you touch the worm anywhere on the posterior part of the animal, it will become activated. And then it can send signals either through a chemical synapse that's located down here on this ventral branch, or through electrical synapses. And there's one cluster of them right in the proximal axon. And there's another cluster of electrical synapses right at the tip of the axon. Now, although all of our experiments are done using C. elegans, what we're really interested in is understanding how normal patterns of axonal connectivity can be disrupted in neurodevelopmental disorders. So by way of example, on the left side, you can see a depiction of the corpus callosum in a normal control child. And on the right side is an example of the corpus callosum in an autistic child. And you notice this is both disorganized as well as being underdeveloped. Now, just another example here, this is a mesolimbic pathway shown in red. There's a typical child on the left side. On the right side is an example of the mesolimbic pathway in an autistic child. And you notice this is very much underdeveloped. So really our interest is to try to figure out how genetic variants that have been associated with neurodevelopmental disorders, how can these genetic variants disrupt normal patterns of axonal connectivity? And to start with, I just give just a brief overview of how these genetic variances are even identified in humans. And this has been really for the past 15 years or so, the real focus has been through the identification of de novo variants. So what happens in these types of studies is that the investigators will assemble trios. So the trio includes both a mom and a dad who don't have the disorder, as well as a single child who does have the disorder. They then obtain either exome sequencing or more recently, it's probably more common to do whole genome sequencing. And they identify the variants that are in the child, but not in either of the parents. And then from there, what they do is they look for genes that are actually accumulating these de novo variants at a rate that's greater than expected by chance. And what you see here is just a graph. This is published a few years ago, and it's showing you, all the genes that have been identified to that date as having genome-wide significance with various neurodevelopmental disorders. So my talk today is going to focus on two ongoing investigations in my laboratory. The first one involves variants in CACNA1C. So this is the name of the human gene. Now worms would call it EGL19, but at any rate, in humans, the CACNA1C has both common and rare variants that are associated with autism, but also many other neurodevelopmental neuropsychiatric disorders. And I'll show you how this causes disruption of axonal connectivity in C. elegans. And then the second part of my talk is going to involve this gene in humans. It's called RBM27. And de novo missense variants have been identified in a handful of children that include both with autism as well and/or intellectual disability. And I'll show you some of our work in C. elegans, how this actually leads to axon degeneration. So to begin with, starting off with a CACNA1C project. So we actually got interested in this many years ago. It was actually initially with this publication way back in 2013. And this is a GWAS study. And the goal here was actually to identify genes that had significant association with multiple neuropsychiatric and neurodevelopmental disorders. So specifically, they were looking at autism, ADHD, bipolar disorder, schizophrenia, as well as major depression. And what they found from this, and you can see this is a Manhattan plot. So each dot here represents just a single common variant. And this red line represents genome-wide significance. So you can see it was only 4 genes that actually reached genome-wide significance. But what really caught our attention here is that 2 of them, so it's CACNA1C and CACNB2, these both encode subunits of the same voltage-gated calcium channel, right? So CACNA1C encodes a pore-forming subunit, and CACNB2 encodes this beta auxiliary subunit. So what we think, in terms of these common variants, we think they're really interesting because, for example, the CACNA1C risk variants that were identified here are actually present in 1/3 of the human population. So because of that, we think it's going to have a very prevalent, effect in the development of autism, overall. So it's not going to be just affecting a tiny percentage of autistic people. It's probably contributing a much more widely. But on the other hand, in terms of trying to do experiments with model organisms, these are not very useful because, well, one thing is that it's an intronic region that's not conserved. Another thing, they actually have very small effects. So they're really risk variants rather than, a causative mutation per se. But fortunately, in the case of CACNA1C, what other thing that we noticed is that, there had been de novo, so these are rare de novo mutations that have been identified, and they're considered to be causative for this thing called Timothy syndrome mutation. Now, Timothy syndrome, really the worst phenotype there is irregular heartbeat, because that's causing heart arrhythmia, and it ends up being fatal in almost all cases during childhood. But the other thing is they also have defects in their hands and feet. And of course, a part we were really interested in is all of them have severe neuropsychiatric disorders, and the exact disorder varies, but many times it is autism or intellectual disability. So, at the time that we started working on this, really because of this, everybody was interested in the heart. So it had been studied extensively in cardiac cells. And there are actually many different variants. You can see the yellow dots represent the different Timothy syndrome variants, but they're all gain of function. So they're actually increasing the calcium conductance into the cardiac cells. And some of these were found just in one child in the whole world, others, maybe 30, 40, but overall, so far, there's only like 100 or so known cases that have been reported for Timothy syndrome. So it's really, really rare. But on the other hand, we think anything we discover using this variant is going to be applicable to the much broader population of autistic children, just because you have those common variants in the same gene. And by the way, the common variant is apparently also a gain of function, right? So it's been studied in iPS cells, and it appears to also increase calcium conductance, just to a lesser degree than the Timothy syndrome mutations. So first thing we wanted to do, because this had never been studied in neurons, as I said, is just to take a look at the region. So some of the most common mutations are these. So in humans, it's G402R that's here, but also in blue is G406R. These are kind of at least G406R is one of the most common ones. Now, G402R is less common, but our stock center actually just happened to have a mutant that had been isolated probably decades ago. So we ordered that. And what we found is that causes this phenotype where the PLM axon, normally, as I said, it terminates before reaching the territory of this ALM neuron. And what we found in these mutants is that it overextends. So it grows too far and it's invading the territory of the other neuron. And this is one of the mutants, but then we went on using CRISPR to generate several other mutations, and we found that they all had the same phenotype. Another thing we did take a look at is this, in terms of synapses. So as I mentioned, there is that chemical synapse, and that appeared, it's not shown here, but it appeared normal. And then there's also the electrical synapses at the tip of the axon. So we wanted to see what happened with that. And you can see here's the wild type. This little, where there's an arrow, you may be able to see a dot there. That's the cluster of electrical synapses. And that's still there. It's still at the tip of the axon, but it's just displaced too far forward, right? Because of the axon being overextended. So anyway, the synapses are normal. It's just that it's misplaced. So one other thing we did is to look at behavior. So these are touch receptor neurons. So as I mentioned in the beginning, if you touch the worm, it activates this neuron. And the other thing it does is it elicits a response where the worm is going to move in the forward direction. So a sort of escape response. But the other thing that happens is every time you touch it, it's less likely to respond. So it's a kind of habituation. And we found, so the blue line here is the wild type. And we found that, it habituates as expected, but then in the mutants, it also habituates, but it does it really excessively, right? So it habituates much too fast. So that's kind of the work that we had finished many years ago now, and that have been published, but we're now going on. So this is now getting into some of our more recent unpublished work. So we're moving on and trying to get at some of the mechanisms that cause these defects, both in the behavior, but we're actually most interested in the axon termination defects. So one thing that we noticed right away is that the EGO19, we made GFP fusions, and we noticed that it was kind of localized to these punctate spots within the axon, and these co-localize exactly with late endosomes. So we use this RAB7 fusion as a late endosome marker. So that was interesting. And one of the reasons we're kind of interested in this, because it's known that calcium signaling is important in late endosomes. And there's many different channels that can transmit calcium in late endosomes as well as lysosomes. And this, the CACNA1C, the ortholog, or EGO19 hadn't ever been identified there. But there is this other similar voltage-gated calcium channel. It's known as CACNA1A, and at least in Drosophila and also in mouse neurons, this is localized to lysosomes, and it can mediate fusion with autophagosomes. So, just extrapolating from there, we wanted to see if there was any function in these mutants, if they actually affected the late endosomes. And so what we did, we used Rab7 as a marker, and these late endosomes, these are chymographs. You can see the late endosomes are moving, at least a portion. We find about 60% of them are moving normally in the wild type. And you can see that here. This is a wild type, about 60% moving. And then these are two different EGL19 gain of function mutants, and both of them significantly reduce the portion of moving late endosomes. On the other hand, this GLO4 is just, this is another mutant that has the same axon phenotype, but it doesn't, we found it doesn't affect the late endosome motility. Right, so this, this is, as I said, it's unpublished. It's very much a work in progress. And one of the next things we want to start doing is to actually do calcium imaging and see if the mutants create any calcium signal that's associated with these late endosomes on one possibility. The other possibility is it could just be flooding the whole axon with calcium, and that could also affect the motility. So we'll look for that. And also, we can start trying to manipulate calcium levels and see how that affects the endosome motility there. But in the meantime, we also are going ahead and trying to look for genetic interactions. So the way we are addressing genetic interactions is actually just doing RNAi screening against other orthologs of autism-associated genes. And we haven't tested them all yet, but we've tested about 35 or so. And so far, we just found one interactor, and it's in this gene known as SHIN1. So this encodes SHANK in mammals. I mean, it's an ortholog of SHANK. And SHANK has actually been very, very well associated with autism, as is CACNA1C. And it's also been studied extensively for its role in synapses, especially the postsynaptic compartment, but not really at all in terms of like axon growth, axon termination. But anyway, what we did find is that while we have different mutants, so we have a weak mutant, it has no, it's like an in-frame deletion. It has no phenotype on its own in terms of this axon overextension phenotype. But when we make a double mutant with the EGL19, you can see that's it enhances it, right? And then we also, though, have two different null mutants, and both of them cause this axon extension defect on their own, right? And then on top of that, they do enhance the EGL19 as well. So the end result here is that we think that SHIN1 does, it promotes axon termination, but it also interacts genetically with the EGL19 gain of function mutation. So we wanted to go on to kind of address what might, how that might be happening. So one thing we did was look at EGL19 GFP fusions, and we found that it's in the axon, but it's especially concentrated right at the tip of that PLM axon. So that was interesting. But then when we took different, so these red lines are all different EGL19 mutants, gain of function mutants, and we found that all of them are very significantly decreasing SHIN1 localization at the axon tip and also within the axon too. So there's a lot less SHIN1 in those mutants. So essentially the EGL19 gain of function mutant is disrupting localization of SHIN1 in that PLM axon. And then these are just some other mutants. Again, these are all things that cause that axon termination defect, but apparently through a different way because they're not affecting SHIN1 localization. But one other thing that we did do, so there's the other thing here. This is a Rab7 mutant, the red arrow. And Rab7 is needed for normal endosome motility and endosome function. And we also, so we thought, so we just tested that mutant and we find that also affects SHIN1 localization in the axon. So it's disrupting it much as the EGL19 gain of function, but we also treated our worms with chloroquine and that does the same thing. So we do think this kind of endolysosomal activity, the motility is promoting SHIN1 localization in the axon and at the tip of the axon, and the EGL19 is disrupting the localization. But the other thing we wanted to do is ask if we force overexpression of SHIN1, could we actually suppress EGL19 phenotype, so the axon overextension, because the idea is that you're removing, EGL19 gain of function is leading to a lot less SHIN1 at the axon tip and within the axon in general. And this remember, SHIN1 actually promotes exon termination. So we think this might be causative for the phenotype, but to really test that, we're adding extra SHIN1 to transgenic overexpression. And you can see here, the phenotype is suppressed. So EGL19 gain of function causes about 50% penetrance of that phenotype, and then it's reduced to 29% when we overexpress SHIN1. Right, so that's it for my first part on EGL19. So again, that's, all those experiments at the end are just really ongoing work that hasn't been published yet. And then this last part is something that we published fairly recently. So this involves the RBM27. So it's an RNA binding protein. Now, why we got interested in this is because it was listed. So there's something called the Simons Foundation. And they're interested in autism, and they kind of host and sponsor this database that collects information on genes that might be associated with autism. So, and they rank them, like they all have different, some of them have like very strong evidence, some are kind of circumstantial, some, they're all different ranks. Anyway, this one is considered a strong candidate. It's RBM27. And they're really generous and kind of liberal with their rankings, because I feel like, all they're going on is just they found 5 of these de novo mutations in children with either autism and or intellectual disability. And they're all missense mutations, which means you don't even really know if they're affecting protein function at all, because they're just missense mutations. And there's only 5 of them. There's no like kind of statistical analysis that we usually expect, to get a high confidence autism gene. But anyway, that's what they had. And they kind of put that out there, I think, so other people can kind of look into it and test it. But so what we did is we decided to use kind of C. elegans to sort of test this idea. First, is a protein affected? Is it causing any neurodevelopmental phenotypes? So what we did do first is these are the 5 de novo mutations that have been found in people. And we just compared it to the worm ortholog. So it's in worms, it's actually called RBM26 instead of 27, but we compared them and we found that just 2 of them. So it's these 2 here, they're L13V and also P79L. They, those 2 are at conserved positions. So we went ahead and created, we did a few things. First thing is we just inserted a flag tag and then into the RBM26 gene. And then we also inserted these 2 missense mutations. And we found that both of these substantially, we did Western blotting, and we found, this is a wild type normal expression of RBM26. P80L, you see it much, much less. L13V is also much less, but not quite as much less of the protein expression. So certainly these are missense mutations, but they're absolutely having a big effect on the levels of protein. So that's already important, because as I said, one of the problems with even calling this as an autism candidate gene is we didn't know if these mutations would affect, the protein, but apparently they do. And then the next thing we did was to look for phenotypes. And one thing we found is that this PLM axon normally should terminate right there in the midbody, but it does overextend, at least in young larvae. Now we did find for this phenotype by the late larval stage, this phenotype basically resolved. So this kind of, this overextension or phenotype basically goes away as the worm is maturing. But we found this other phenotype we ended up focusing much more on because it just increases and gets worse with age. But it starts in the earliest larval stages, and we see these swelling. So we call this a beating phenotype, and essentially the axon is swelling. And this is something that's been observed within neurodegenerative disorders in humans. And we did find all these phenotypes in like a null allele. So it's just something we got from our genetic stock center. So it's a deletion of the RBM27 gene. And we also found the same phenotypes, just a little bit less penetrance with each of those 2 patient-derived alleles as well. So one thing that we wanted to do then was to see where it's localized. So we just inserted a scarlet tag into the genomic copy of RBM26. And what we did find is it actually in all cells. So it's not just in neurons, but then within neurons, it appears to be concentrated into the nucleus. So you can see this dot, here's like the whole neuron, and where you see the yellow, that's where the nucleus is. So it's concentrated there. Now we do also see it in the cytoplasm, but it's just much fainter. So it's concentrated in the nucleus. But because it's in all cells, we want to know, what cells is it actually functioning in to cause these different, to protect against these different phenotypes. So we use like auxin-induced degradation. So our scarlet tag that I mentioned before, what I didn't tell you is also has a auxin-induced degron, and we can treat these worms with auxin, and essentially we can do this in a way that it's like that is specific to different cell types. So anyway, we do that, and what we did find, if we're selectively degrading it in neurons, we see both of those phenotypes. So we see we see the axon overextension phenotype, but we also see the axon beating phenotype. But when we do it in these other 3 tissues, we don't see that phenotype at all. So it's the same as the controls. So anyway, that's kind of telling us that it's working in neurons. But we did need to test the same thing in another way. So we did this through transgenic rescue experiments. And here, what we found is that we could rescue both of these phenotypes. So there's the overextension phenotype as well as the beating phenotype. We could rescue both of those through, by specifically expressing the wild type copy of RBM26 in neurons, right? So and specifically we did it like in touch neurons. So it's the PLM and a handful of other neurons. So we're able to rescue those. So at any rate, the point is that this is a cell autonomous defect there in phenotype. So another thing that we wanted to do is to take a look at mitochondria. And our reason for doing this is simply that because we had this this kind of neurodegenerative phenotype, and we do know that mitochondria are very commonly implicated in neurodegeneration. So we took a look at the density of mitochondria in these axons, and what we did find is that it's diminished, both by the null allele, but also by both of the patient-derived alleles. And we did this at different stages. So it's like first larval stage, second larval stage, third larval stage. And anyway, we found at all these different stages that there's less mitochondria. So you can see the mitochondria here. They're shown with the little dots here. And this is our wild type, and that's the control. So there's a lesser amount of mitochondria. But we also wanted to look at the kind of a marker for mitochondrial health. So this is oxidation marker. So essentially what we think as mitochondria age, or if there's something wrong with them, the electron transport chain is not really working correctly. There's a lot of reactive oxygen species that are released, and it causes oxidation. And we can detect that using a mitotimer sensor. And that starts out green, and then it gets oxidized. So it's going to turn red. And you can see here, this is our wild type, and it does oxidize over time. So as the worms get older, it's getting more and more oxidized. But the thing is that in our RBM26 mutants, it oxidizes much faster. So you can see that it's getting more red than the controls are. I mean, we tested this again at all different larval stages, and we found that at each stage, although the controls are getting more and more oxidized, the RBM26 mutants, at least we tested both of the patient-derived alleles, both of them are getting more oxidized and faster. Okay, so next thing we wanted to do is try to address the mechanism. So we want to ask, we do know that this is affecting mitochondria, it's causing degeneration. We really want to know why. Now, since this is an RNA binding protein, then our goal was just to find RNAs that are bound, that it's binding to. So the way we did it, because the worms are completely transparent, so you can just shine UV light onto them, and UV is really, really good at cross-linking RNA and protein. So we did that. So you shine the light, and that'll kill the worms, like when you're doing it, but then you can immediately lyse them. And then we can use our flag antibody to pull down the flag tag version of the RBM26 protein, and then just sequence all of the RNAs that come with it. And there were actually we were surprised at how few we found, just about, I think it was about 64 that reached statistical significance. And looking at those, they're actually all different kinds of RNAs that you can see here, like the pi RNAs and SNO RNAs, et cetera, and also mRNAs. And we found, for the time being, we're just focusing on the mRNAs, and we found about 24 of those. Only half of them actually had human ortholog, so we focused on those 12, and the best p-value there was MALS1. Not only was it the best p-value, we're also really interested in it because it's a mitoribosomal assembly factor, or it's an ortholog of what's known to be a mitoribosomal assembly factor. So this hasn't been studied before in neurons or in worms at all. But the idea of what it does in human cells is that it acts as a brake on mitoribosome assembly, right? So as you know, the mitochondria have their own special ribosomes, and what they do, they just translate everything that's encoded within the mitochondrial DNA, which is just a small, like a handful of genes, which are really just certain components of the electron transport chain. But anyway, this is an assembly factor because this mitoribosome consists of both a large subunit and a subunit, and there's more than like 100 different components. So it's a very kind of long and complex assembly process. And what happens is this ortholog, so the ortholog of MALS1, it's actually known as MALSU1. It binds to the large subunit during the construction process. So while it's been constructed, it binds there, and its job is to prevent premature assembly with the small subunit, right? So once this large subunit is done, it actually dissociates, and that allows the small and large subunit to come together. So it's essentially just a brake on assembly. So one thing we wanted to do is just to see where this is localized, the MALS1, and we actually found that it's localized in axons it's localized in the whole cell, of course, but within axons, it's specifically co-localized with mitochondria. So we have mitochondria shown in green, and this is the MALS1 scarlet fusion protein. So it's exactly corresponding. And this is something, again, it hadn't been studied in neurons, but within like other kinds of mammalian cells, they did see this same type of corresponding. So it's MALS1 is specifically within mitochondria. So then we went on to ask, so what I told you, of course, RBM26 is binding to the MALS1 mRNA, but we want to know what is it doing with it? So how is it affecting it? So we checked both with protein and the mRNA. So we used qPCR to check the mRNA expression. And we did find that in our two different RBM26 mutants, we see a big increase in the amount of mRNA. Right. So, and same thing with the protein. So this actually at the protein, when we did our normal Western blot, we couldn't even see it at all. The mouse couldn't detect it. But then in both of the RBM26 mutants, you can detect it, and it's a lot, it's quite a huge amount more than wild type. And then if we did play around with some of the conditions a little bit, we could eventually detect it as very faint bands. But again, it's much increased by both of the different patient-derived RBM26 mutants. So taking like all this together, it's just suggesting that RBM26 negatively regulates both MALS1 mRNA and MALS1 protein. Now, of course, that's interesting, but what we really want to know is whether this excessive, because again, it's a huge excess of MALS1, could that actually be responsible for the phenotypes? So we tested that in two different ways. One is just by forced overexpression of MALS1 using a transgene. And we did find that causes, when we did that, we tested two different transgenes, and it does cause three of the different phenotypes. So it affects, it does cause that axon overextension phenotype. It also causes the beating phenotype, and it also decreases the density of mitochondria. So that was one thing, but another thing I think really in many ways more important was to ask, can we suppress this phenotype, right? So in the RBM26 mutants, there's way too much MELS1. So we want to take away all that excess MELS1 using a double mutant. And what we see here is, so you can see, and this experiment really worked best with the beating phenotype. So you see that in panel A. So this bar right here is RBM26 single mutant, or one of the single mutants. And then this bar over here is a double mutant with MELS1. And we tested multiple RBM26 mutants and multiple MELS1 mutants. But either way, we found the same result each time. And it's actually completely suppressing the phenotype. So it's back down to the wild type levels. So it's essentially completely curing that phenotype. Now, we also found a suppression, at least as a mitochondrial density, just not as substantial. And it's actually not on this slide, but for the other phenotype, the axon overextension, it actually didn't suppress it at all, but that's because the MELS1 mutant causes that phenotype on its own, and it doesn't suppress it, but it doesn't increase either, right? So it's also, it's also consistent with these things acting in a pathway. This is kind of what we think might be happening here, is that, as I said, MELS1 acts as a brake on mitoribosome assembly. So on the left side is what's happening normally. So it binds to the large subunit, then it dissociates and allows the subunits to come together. But we think if you have way too much MELS1, what's going to happen is that this is not going to really be able to dissociate so effectively. And that's going to interfere with the normal formation of mitoribosomes. And, in doing that, because these mitoribosomes are really important for the electron transport chain, that's going to affect the mitochondria function, right? And that's why we see that excess oxidation, and we also end up seeing that degeneration phenotype. So this is just a summary of kind of what, these results. So we think that normally RBM26 negatively regulates the MELS1 mRNA, but then if you lose RBM26, so in those mutants, you're getting way too much MELS1, and that's of course going to cause way too much MELS1 protein, and that affects, as I just showed you, we think it would affect the mitoribosome biogenesis. So that's, that's going to lead to dysfunction and axon degeneration. So the other thing that I can share with you, this is, just kind of something we read about in the literature. It's not anything that we've done, and it's just hypothetical. But, our genetic results and our biochemical results do show that RBM26 negatively regulates MELS1. We don't know exactly how it does that, but this RBM26 protein hasn't been, in worms or in humans or any of its orthologs in any animal. There's only 2 papers published on it, and they're both in HEK 293 cells, I think. And the point of those papers is just that they discovered it as being a part of this thing known as the RNA nuclear exosome, the nuclear RNA exosome, and it's actually here. So this is RBM27 right here, and it's another. It's actually 2 different genes, it's 26 and 27, but they think that both of these are part of this RNA exosome. And what this RNA exosome does, it's a nuclear RNA exosome. So it's in the nucleus, and it's kind of thought to be involved in surveillance, and it can degrade RNAs. So that would make sense, given that we found that, this is negatively regulating both the mRNA and the protein. So we think that this might be kind of explain that negative regulation. But again, it's just hypothetical at this point. But the other very interesting thing, though, is that different components of this, so there's many components you can see shown here for this RNA exosome, and most of these components actually have been associated with this particular type of neurodevelopmental disorder. It's called pontocerebellar that is spelled wrong, but it's hypoplasia, I think, type 1. Yeah, so it's pontocerebellar hypoplasia type 1. And also there's some other related things to that, but they're all associated with de novo mutations in the genes that encode this. And so we think that probably these de novo mutations in RBM26 are maybe just part of this syndrome, really. So that's what we're thinking. But again, this is just hypothetical. So finally, I'd like to thank well, the members of my lab who did this work. So the first part, when I talked about CACNO1C and its ortholog EGO19, that was done. That was a doctoral thesis of Tyler Buddell, and then Haley Berryman's master's thesis is kind of that unpublished part that we're still working on. And then Tamjid Chowdhury is responsible for all the work on RBM26. Yeah, and then also thanks for, I have internal funding from University of Wisconsin-Milwaukee, as well as the Greater Milwaukee Foundation, and then both NANDS as well, more recently from NIMH. All right, thank you. All right, any questions then? Yeah, Claire.
Have you looked at all for circumstances in wild type that change the amount of RBM26 or NAL1 or nuclear exosome abundance or activity?
Oh yeah, no, we haven't done that yet, but I think it's, it would be a good idea to look at that. So are you thinking about like aging or?
Yeah, if this is a point of regulation for like cell growth or
Yeah.
a way to adjust metabolism.
Yeah, so we haven't, yeah, so it probably, I mean, that RNA exosome is really important just with the homeostasis of RNA. So, I think that RBM26 must be involved with it, but it's just not anything that we've looked at yet. But yeah, it should be, and it would definitely be something that's interesting. Kind of work that, so we're kind of extending that RBM26 work now. Still like very, very preliminary, but we think the mutations actually have a separate set of phenotypes that show up in aging worms. So that might have something to do with that, but we're still looking into that. Yeah. Yeah.
So with looking at the different tissue-specific and cell-specific effects of RBM26, have you, are there promoters that might be specific to the PLM neuron specifically, or maybe, I think you said you were able to overexpress in just a small subset of neurons?
Yeah.
Have you tried doing the AID experiments in more specific areas other than just the neuronal tissue?
Yeah, so the only promoters we have, they're actually active in 6 touch receptor neurons. So that's about as specific as we can get at. And the AID experiments actually weren't even that specific. Those are targeted against all neurons, just because of the available reagents and strains that we have. But yeah, I mean, so does that answer your question?
Yeah.
Yeah, all right. Yeah.
So once you found all these structural changes, which are really interesting, do you have an inkling of how these translate to behavioral phenotypes?
Yeah, so that is something that we've proposed to look into, and we haven't actually gotten very far with it yet. So the behavioral phenotype, so for RBM26, we haven't even looked yet for behavioral phenotypes, but with the EGO19, there's, we see that increased habituation, and there's many different possibilities of how that could happen, and we don't know which one it is yet, because we don't even know, it could be even happening within the PLM. We don't know if that's cell autonomous yet. It may or may not be. But anyway, what I'm getting at is one possibility is that, so it turns out that calcium is actually important for that response, for that touch response. So it could just be having extra calcium is enhancing the response. Or it could be, another idea is that electrical synapse is misplaced. Right. And one thing we did look at is, where is it being misplaced to? Well, it's still connecting to the same axon, but it's too far forward. So that could change, the way the information is processed. So there's actually a whole network of, it's called a touch receptor network, and it's a whole bunch of neurons that work together to generate that response. So it could be affecting that in that way, or something else that we can't even imagine yet. So yeah, it's something we want to look into, but we haven't gotten that far yet.
This is a bit of a stretch from C. elegans to humans, but like, so C. elegans said it makes them sensitive to touch. Is that just that maybe it corresponds to the opposite trait of being like touch sensitive to different textures? Or
Sorry, I'm sorry, I couldn't quite hear what you were saying.
Curious if, so C. elegans, you see that it results in this behavioral phenotype of touch. Does that suggest that maybe in humans, maybe this is responsible for like the autistic trait of being touch sensitive or sensitive to different textures?
Yes, well, so actually the results that we got are kind of opposite of what you might think in terms of thinking with humans, because we found enhanced habituation, but for the most part, it's generally thought that autism causes like a lack of habituation. So it's actually the opposite. But I've also talked to a few people about that question. They said, well, it's not necessarily always the case. In some cases you might see increased habituations in certain aspects in autism. But really my opinion on that is it is too much of a stretch. So I'm happy just to have a behavioral defect. I don't know if whether we can really connect that to what's going on in autism. Yeah, like our emphasis is really just, how does it affect the axon development? Yeah, but thanks. Right there. Yeah.
I was curious, you saw the defects in mitochondria and the axon. Did you also look in the cell body to see if you saw similar?
No, we haven't done that yet, and it's just that there's so many mitochondria there. I'm sure you could do it. You probably need like a better microscope than what we had, but yeah, we haven't done that. But although, come to think of it, I guess we have done it because in terms of the density of mitochondria, we couldn't quantify that in the cell body, but it turns out that the oxidation sensor, so that was the opposite problem. The oxidation sensor, so we can't detect it in the axon because it's so faint, but we could detect it in the cell body. So apparently it does affect mitochondria in the cell body. Yeah. Yeah. Go ahead.
I was curious, earlier you were talking about how you were determining RBM26 is for all different cell types, and you said that you used auxin to like differentiate different cells. I was wondering how that works.
Oh yeah, so sorry, I had actually kind of glossed over that a little bit, but the way it does work is that you use tier 1. So tier 1 is so auxin, I think is a plant hormone, right? So and tier 1 is a plant protein. It's an F-box protein. And what it does is tier 1 degrades any protein that has this AID, which is an auxin-inducible degron, right? So what we do then is just we use cell-specific expression of tier 1 because worms don't have it because it's a plant protein. So it doesn't exist. So we can put it into whatever cells we want. So we have different, so that we just get from the stock center. It's another lab generated it, and there's many different, we call them driver lines, and it drives expression of tier 1, like in neurons or in hypodermis or gut or pharynx or muscle, whatever it is you want. And then we just cross those with our degron tagged RBM26 gene. And then the next thing you have to do is to add auxin to them. So the auxin is nice because it's you can use it as a temporal control, right? You can have auxin or not have it. And then cell specific depends where you're expressing the tier 1. Yeah. Up there.
I have 2 questions. First, for the legal gain of function mutant, you see that there's decrease in habituation compared to the wild type. So the decrease in habituation.
So it's actually more, it's extra. Yeah.
That happens over time. The rate at which it happens, is it similar to that of wild type?
So yeah, they're both habituating, but the mutants habituate faster, right? So there's they both habituate, whether it's mutant or wild type, but it's just happening faster. Yeah, and there was one other thing related that I just didn't go into it, but and this is published work, but one thing we found is that we have this selective autophagy gene, and that it's called WDFY3. And we were really interested in it because it's also an autism associated gene. And we made double mutants, and we found that completely suppresses our exon overextension phenotype. The interesting thing, it also completely suppresses that habituation defect as well. So yeah, it was on that graph, but I just didn't get into it. So.
Second question is that in the one RBM26 mutant, since mitochondrial dysfunction goes up with age, do you think this RBM26 mutant animals, which also leads to mitochondrial dysfunction, could lead to mitochondrial dysfunction, have an effect on the lifespan of the animal?
Yeah, so that's a really good question. And it's funny because I just asked that question to the person who was doing this work. And he thinks it does, but we don't know yet. It's kind of, so what we did, and the reason he happened to just observe that, because as I said, originally, so in our published work, we were just looking at this degeneration phenotype. The interesting thing about it is that it happens, we see it in the earliest larval stages. So it's like an early development. But another interesting point I didn't get into is that in those that human syndrome, the PCH1, that's axon degeneration that's happening in infancy. So we think there's, kind of a correspondence there. But more recent, we never looked at this before, but we've now found that there's a whole other set of phenotypes that are happening during aging. And because of that, we never looked at older worms. But anyway, we see those, but at the same time, we think that probably they're dying quite early, but we don't know that yet. That's our hunch at this point. Yeah, you're welcome. Yeah.
Maybe you said this and maybe I didn't catch it, but are we going through all these analysis and findings so far? Do you have a hypothesis for the reason why the proximity of the two axons might probably cause an effect as opposed to
Oh yeah, so why the overextension is causing are you asking about like the behavioral phenotype then? Yeah, so why, and that we don't know, and it's something that we've looked into. It could be because it's changing the location of an electrical synapse. That's one possibility. It could just be because there's a lot of extra calcium in those neurons, and because we do know calcium is actually involved in that touch sensor system. So we don't know yet, but those are some possibilities. And it could also, we don't even know if that effect is cell autonomous. It could be EGO19 is affecting some other neurons within the touch, because there's a whole set of neurons that are interconnected, and that's all part of this touch response circuitry. So it could be in another neuron, as far as we know. So it's something that needs a lot more investigation. Yeah. Yeah.
Would you mind pulling up that touch sensitivity habituation graph?
Oh yeah.
I guess what I was wondering, so you see the huge difference between wild type. There we go.
Yeah.
But it looks like it's that very early. It's the difference between 1 and 2, and after that the graphs seem to follow basically the same slope. Right?
Okay, so what are you?
For why is the touch number between 1 and 2 touches is so radically different, but then yet the percentage of habituation thereafter levels up?
Yeah, I mean, that's a great question. Not anything I could actually answer, but it's,
is the molecular mechanism of that habituation well understood?
It's not. Yeah, I mean, that's part of why I can't answer it, because the molecular mechanism, we just know that it has something to do with calcium, and apparently that's just what's happening. We don't know why, but that is a good observation, that it's just the second touch that's really, really strongly affected. Oh yeah, up there.
I was gonna ask if you thought about looking at the mechanosensors like expressed in the PLM, and well, this is interrogating the PLM neuron function, right?
Yeah.
So, do you think they could be affected by?
Yeah, I mean, that's definitely another good hypothesis on how this could be happening. Again, it's we just haven't had really, we haven't looked into it too much yet because it's not easy to do it. But yeah, it's a good hypothesis though.
My other question is, there's a lot of very easy to quantify behaviors in C. elegans, and I was wondering if you've noticed any differences in other behaviors, like there's a defecation motor program, also like how the tail moves, controlled by the DA9 and DA8 neurons.
Yeah.
Have you noticed anything?
We haven't noticed, but I can't say that we've really specifically tested or quantified anything. So, yeah. So again, we didn't know, but it would be interesting to look into those. And then if we did that, also to see if, other neurons are being affected, right? So whatever neurons are responsible for those behaviors, we would want to look at those as well. But so far, we're just concentrating on this one neuron. Yeah. All right, great. If that's it, thank you very much. It's been a really great visit here today. And again, to thank Claire for organizing everything. Yeah.